I'm thrilled to talk to you today about data-driven methods. This is a chapter in our lab's work that is focused on data science applied to synthetic biology. I think there's tremendous opportunity and potential of applying data science to synthetic biology, and I'd like to show you two successful vignettes of data science applied to synthetic biology. So we're going to be talking about designing biosensors, in a loose sense of the word, biosensors, and then identifying genetic targets for biological control in non-model microbes. And why non-model microbes? Well, non-model microbes are becoming increasingly prevalent in synthetic biology. I think the field at large, I think, Tom, it's safe to say this, correct me, you know, if he's more experienced in this, but I think the field is at large is moving towards working with extremophiles, halophiles, acylophiles, organisms that live in these extreme environments that can perform some useful function that we would really have a hard time trying to like coerce E. coli or Pseudomonas or Bacillus to do. There's sort of, you know, certain built-in features that these microbes have. So with that, though, comes the baggage of not knowing what the microbe actually is and how it works and the genome and so forth. And so that oftentimes is an opportunity to look at biological systems in a new light. Instead of relying on 50, 70 years of E. coli research and looking at all these past papers written, right, you have to try to quickly bootstrap with the tools that you have. And what do we have nowadays? We're really good at artificial intelligence, right? At least we hope so. And then secondly, we're also really good at data collection. So this is why we're interested in data-driven methods. Now, the three themes of my lab I call it the biological control lab. So the three themes are control. I'm interested in how to use genetic circuits to enact feedback control. I'm interested in DNA-based control, supercoiling-based control, enacting networks and topologies that give robust the phenotypes. I'm interested in computations. I'm interested in understanding how biological processes generate a certain type of phenotype or signature. So here you can see a panel of the biosensors that we are going to now talk about today, and they're all responding with these crazy nonlinear functions. And the first time the professors that were on the committee, the PhD committee for this student, Akib, saw this plot, they said, wow, that's a great simulation. And it's like, those would be great dictionary functions to use in a nonlinear approximation problem. And it's like, no, those are actually experimental data. And those are all individual strains that produce a wide range of response to a particular chemical. And the last aspect, I'm very interested in adaptation and learning. You can think of this as also as the theme of robustness, but basically I'm very interested in understanding how biology responds to chemicals and modeling that response model, understanding like what are the phases of response? Can we think of it as a layered process? How do we back out and tease these things? So at the core, there's a lot of network reconstruction problems that I'm interested in. Network reconstruction was a research question that was very popular in the early 2000s. But you know, sort of with new legs, the legs of machine learning, and of course all of the complexity that comes with overfitting and underfitting and model capacity. So, so here's my talk today. I'm going to talk about two parts, and then I'm going to give a brief teaser to some of the new projects that are starting in our group. The first part, and all this work credit goes to my two amazing students. Absolutely, am so grateful to have Akib Hasnain and Shara Balakrishnan as my first two PhD students. They graduated just this last June, and they are on to exciting new things. Akib is going to be a postdoc at Lawrence Berkeley National Lab, and Shara is going to be a CTO of a startup that's managing luggage, finding your luggage when it gets lost at the airport. He's using machine learning and control theory. And so two really brilliant students, and they are totally responsible for all the amazing results in this work. And so in the first part, I'm going to talk about a completely data-driven way of ranking genes as candidate biomarkers in a non-model organism. So how do you, there's a little bit of math. Raise your hand if you have a background in quantitative background somewhat. Thanks, Guy. Jose is like half. Yeah. Not really. Okay, so there's going to be some scary math. I apologize. Okay, you guys can ask all the questions you want. All right, because this is even more scary math. So you want to like make sure you understand that, and then if we get to this, that's great. You know, if we don't get to it, it's not a big deal. All right. So And then we're going to talk about the synthetic biology, which is actually quite straightforward: extraction, construction, and characterization of these organophosphate sensors that we get from this biomarker process. And then if we have time, I'm going to talk about a totally new set of results that are very exciting. It allows you to characterize the principle or the property of observability in a biological process in a data-driven way. Nonlinear observability has historically been a really hard thing to write down and to compute and to analyze. And we've shown that you can actually use it to identify genetic targets for CRISPR repression. So if you want to do biomass control or fitness control, it actually is a way of doing things in a data-driven fashion. Okay, so first, the big picture. I think one can argue that that if you look at the average agricultural setting in the U.S., we have a very strong propensity to use a mixture of organophosphates and generally toxic compounds to control our pests and our fungicides, or our fungus in our plants. And those chemicals oftentimes will pass through the upper half of the plant structure into the rhizosphere and then diffuse into the soil and into the soil microbiome. And we don't really have good principles to understand how to model that, what the effects are, what are the salient sort of indicators that this is happening. And the amount of pesticides that we use in the U.S. is really staggering. So, I know outside of the U.S., like, because I grew up in Hong Kong, like, you can rail on the U.S. as much as you want. So this is an example. Like, we use 1 billion pounds of conventional pesticide each year in the U.S. for weeds, insects, and other pests. And a river that's close to us, that's not really a river anymore, at least nobody wants to swim in it, is the LA River, the Los Angeles River. And there's, for example, 1,556 species in the LA River alone, not including bacteria that are affected by all of this runoff. And so one question we wanted to ask up front as an application-driven question, but which I'm going to then distill into a scientific question, was can we engineer or can we identify a non-model microbe that lives in the soil that is able to persistently respond to a chemical of interest, to some pesticide, an organophosphate compound? And can we track the signature of that response as a generalized notion of cell state? Cell state, right? So the state of something is like the change of coordinates or the change of variables that happens when a perturbation is applied to a process, right? And so cells are these microscopic creatures that have lots and lots of things happening inside of them. And so the question is, can we track that and then see like if there's a consistent pattern in how things are fluctuating, so we can then generalize or abstract a notion of cell state in response to the organophosphate. So we're going to run a very simple experiment. Oh, and here's the summary question. Do the natural transcriptional dynamics of existing soil microbes reveal pesticide activity? This is actually quite an ambitious question. It's quite a risky question as well to give to a PhD student. So, in retrospect, I was probably not that great of an advisor. But Akib is amazing, and he triumphed nonetheless, because this is the question, is like, the answer could just be no. The microbes don't respond at all to the pesticide, right? But the bet that we made was that a pesticide is generally a toxic compound, and so it's going to elicit some sort of oxidative stress or some sort of mutational response in the microbe. And so we want to see if there's some generalizable signature that we can extract from that. Okay. So here's the mathematical version of the same question. All right. So this is the sort of the experimentalist way to ask that question. The mathematical or quantitative way to ask that question is can we use observability theory, classical observability theory, to design novel data-driven pesticide biosensors? Okay. So here's the experiment that we're going to run. We took a non-model microbe for which we know that there's some hope of doing some genetic engineering. We didn't just take any non-model microbe, because if some of them are very genetically recalcitrant, they won't transform. You have to use conjugation methods, and you're getting like 1 CFU for a microgram of DNA, so not very efficient. But we picked Pseudomonas fluorescens because it lives in the rhizosphere of sugar beet plants. This was a soil isolate from that sort of a setting. It colonizes soil, water, and plant surfaces. And furthermore, we wanted to test then its exposure to a pesticide that it typically might see in its life cycle, but that isn't supposed to directly affect it. So for example, malathion is an insect spray that you use to control cabbage butterflies. Cabbage butterflies are those little white butterflies with those black spots that fly around and create all the little holes in your broccoli and cauliflower every time you want to start a garden. And then you finish, and you say, I can't seem to garden well because the cabbage butterfly caterpillars are just eating up all my brassica plants. So malathion is a chemical that's used to control it. It does it extremely well. In the environment, it shows up at about 1.3 micromolar concentration. And so that's the concentration we're going to use, basically a contaminant level concentration. The actual concentration when you apply it as a dose to the plant is much higher. And then what we'll do is just a very simple experiment. We're going to run 2 tracks. On the upper track here, we're going to have a treatment group. And on the lower track, we're going to have a control group. The treatment group is going to be exposed to the chemical. And what we're going to do is sample every 10 minutes the culture from a shaking beaker. All right. And we're going to then collect the cells and freeze them. All right. And then once we froze them, take all of those frozen samples and then extract the RNA out of those cells. OK. So to do this actually does require some customized hardware. If you look for vacuum manifolds that can extract cells quickly, you'll see at most like 6 outlet vacuum manifolds, and they cost like $6,000 for this little shiny metal vacuum manifold. So we engineered our own in the machine shop. We used an extruded aluminum block, and we fastened. We had 12 to 24 fasteners, and then we added these Delrin adapters that had little holes in them that were bored in so that way it could draw vacuum. And then we would basically take, the protocol takes 0.45 micron filter paper and drops them on top of these Delrin adapters. And then you drop the samples as you're running the experiment, one after another. And as the vacuum is continuously engaged, all the cells collect immediately on the filter paper and they can't go any further. You take the filter paper as soon as you sampled it, which is almost instantaneous, and then you drop that into an Eppendorf conical tube, a 50 ml conical tube, and then you freeze it in liquid nitrogen. So the transition from like the time point you're trying to measure in this picture to the actual freezing step where you've frozen it is about like 1 minute. Okay, you sample, it drops in, and 1 minute it's frozen. Why do we need this? Because we want to capture the transcriptomic response. mRNA dynamics happen fast, right? And we want to capture it with high enough resolution that we can see dynamics in the way that, Guy or myself would get excited about, right? The control theorists would say, hey, this is actually a nonlinear system or it's a linear system, okay? So, and then of course, once you have all those RNA, like all of the RNA samples in the cells, you then do an RNA extraction day. It takes like half a day to do that, and you just process a bunch in a batch, and then you do library prep for next generation sequencing, right? And then you do your next generation sequencing. So that's how it works. This is our data. This is like 500 of 6,000 genes plotted as a function of time. Here on the x-axis, you can see the time points. On the y-axis, you can see the genes, okay? And you can see here fold change from 0.5 to 2.0. So there's definitely some systemic response to these. And so now we want to, beyond just looking at a heat map, actually extract out some modal dynamics. And so for this, we use an algorithm that you guys might have heard of. It's taken the world by storm. It's really popular. It's called dynamic mode decomposition. Dynamic mode decomposition says we can think about the fold change response as roughly a locally linear model. This is not a new concept. This is actually a very old concept. But the key is that they want to really focus on trying to extract the modal dynamics associated with an eigendecomposition of a matrix. So a linear model says my transcription dynamics evolve according to some simple equation like like this. ZT plus 1 equals a matrix K times ZT. That's a linear model. K is a matrix. We're looking at ZT propagating forward in time. Okay. But the dynamic mode decomposition really focuses on trying to, instead of estimating the K matrix, estimating the eigenvectors and the eigenvalues that compose that system response directly, all right? And it uses a singular value decomposition algorithm. It does some projections, so that way you get rid of unnecessary modes that are not going to tell you much. But basically, you get back dynamic modes or eigenvectors that define invariant dynamics of your system. You get back the eigenvalues that quantify to some degree the power of those modes. And then the amplitude of the initial mode is then characterized by this this coordinate B. Okay, and so we're going to be looking at fold change. Note here we add an offset. We took our malathion condition and we normalized it by the control condition. And the purpose of that is so that we don't have like anything divided by 0. Because if you divide the control condition and there were some genes that were like 0, then it's just going to blow up to infinity and that'll give you a non-number. So we also filter out genes with low read count and static dynamics, because those genes are going to be the ones that basically, you know, they're not really responding anyways. So yeah,
What's your offset 1?
And our read counts are usually integers, and so 1 is the smallest increment that is like basically noise. Yeah, and read counts, we use TPM, transcriptional reads per million base pair reads, or transcriptional counts per million base pair reads, and that typically comes out on the order of tens to hundreds to thousands. And low read count for us is lower than 40. If it's 40 or less, then we just usually throw it out because we think it's just noisy. And if you plot the distribution of data, it doesn't look like anything. It just looks like things bouncing up and down. So that's the floor of the measurement assay. So yeah, any other questions? Yeah, Jose.
I have a technical one. Most of the genes have some sort of a spurious activation in the bacteria, like base pair possibilities. So give us a feeling of how many hits you need to.
Yeah, so if you just take like if you take a control condition by itself without malathion, you will see genes that just turn on and activate. And if you add the malathion, you'll see genes that differentially respond, right? But then some of them will just shoot back down. And those ones are very difficult to actually like infer anything from. So normally, if you wanted to rigorously tell using just say static time point sampling, suppose I only had 2 time points, okay? You need lots of replicates. So that way you can show that the distribution of data points from the malathion-induced condition versus the non-malathion-induced condition has truly shifted. This is the paradigm of DE-seq, differential expression analysis, right? DE-seq2 is a package that's in R. It's like a standard bioconductor package that you use just to process data like that. But it requires lots of replicates, and it assumes you're taking a few number of time points. Our approach is a control theorist one. We're saying I want to take high resolution time points. I'm not going to take too many replicates because I'm looking for continuous curves, trends. If I see bumpy data, I just toss it out. Yeah. So that is then conclusive, basically. So yeah. Does that answer your question? Yeah. Any other questions? Okay, so here is then the modal response plotted out. So this is the slide that actually gets a lot of Koopman operator theorists excited because people have done this for a long time studying fluid dynamics. So look at velocity profiles of fluids flowing past like a blunt body or something like that, and they show that you can look at data of velocity profiles and then train a Koopman operator model and then plot the modes, and you'll get back these cool like waveform structures that look just like what you see if you were to color your liquids with like dye or something like that. So these are so-called nonlinear structures. So it's very exciting to them. This is the first time doing this in synthetic biology with a gene network, all right? And what we see is that we actually can partition the genes into stable, unstable, and oscillatory modes, all right, in response to this malathion inducer. So the stable modes, what is a mode here? A mode is a superposition of many, many, many genes that when you combine them together basically gives you something like a flat line, okay? An unstable response is a superposition of many genes that when you combine them together, all together still produce a very dynamic or transient response. And then the no response mode here is just collections of genes that when you average them out, essentially don't do anything, okay? Okay, so those are the modes. And what I like to do now is actually then select some of those genes to act as indicators of malathion. All right, I have an organism. I know it's now responding to this chemical, all right? And normally what would you do manually? You would just look at the plot and say, well, I'm going to pick this gene, and this gene, right? This is a quantitative way to do that, all right? So you can just like run an algorithm and then it spits back for you the names of the genes and then you go and find the promoters and then you build your biosensors, okay? So what we do is we say we need to try to maximize the output energy that we would get if we were to down select a subset of the genes. So to do that, if Z of T is all the vector of all the genes, I need to down select for a W matrix that's just going to project high dimensional to a few genes, okay? So we aim to maximize this energy then by looking at the 2 norm squared of YT, which when you write that out gives you this expression in terms of the original system dynamics. Because this is a discrete difference equation, Z0 times powers of K gives you ZT, okay? And so that is this expression right here. Then if you want to map that to the output, you write W multiplied by that thing. So if I take Y transpose Y, I'm going to get basically K to the power of something multiplied by W transpose, WK raised to the power of something. You with me on the math? More or less? Not really? So, it's not necessary to understand where that math directly comes from, but the point is that this is this objective function is just saying I want to maximize the 2 norm squared of YT, which is the energy of YT over all possible time points, okay? So I want to maximize the area under the curve for a sensor that I would select, okay? Subject to a constraint that I'm not just going to have an infinite number of sensors. I'm going to like have some finite bound. Because I don't want to ask someone to build 100 different sensors, okay? So when we plot out, so we could solve this problem. This is a standard optimization problem. It's a quadratic optimization problem in W. And so this is actually very solvable. And once you solve it, you can actually then plot out the different values for W. And when you do that, what you see is that different genes, all right, have different levels of contribution to your malathion response, okay? And in particular, there's a subset of genes which have an exponential contribution. They have a way larger contribution to the response of the cell, okay? And then there are other genes that have essentially very little effect. They're essentially like noise. Now, one of the things that's interesting, so I presented these results to Andy Ellington a while back, and he's like, can you tell me, like, do these genes actually reconstruct the state of the cell too? Like the whole state of the cell? And that's one of the beautiful things about observability. So as a separate fact, when you try to optimize this matrix right here, you are actually trying to optimize the capacity to take an output Y that infers Z. That's a well-known fact from control theory. We don't have to derive it here. But this is so what we're doing is effectively trying to find genes or biomarkers that are capturing as a summary the entire cell state. Okay, you can see that through this plot here. The green genes that have really high sampling weight actually show up as having higher reconstruction accuracy relative to the orange and the blue echelons. And you can see then that this is actually directly correlated with state reconstruction accuracy, right? Okay, so what's interesting though is that when we were submitting this paper for review, people were like, well, why don't you just use DESeq? Right, that's sort of the standard question you ask. And the answer is because it finds totally different genes. DESeq, again, is built so that way it assumes you have a few number of time points and you're just looking at steady state response or stationary phase response compared with an initial time point. This is looking at dynamic response and trying to find like something that wiggles fast or that wiggles with a lot of energy along the way. And so what we found was that actually there was some overlap in terms of the genes that were highlighted using both methods. But on the blue is plotted here our method, the observability maximization technique, and this beige or orange color is the DESeq gene. So it actually complements DESeq. It finds different genes that are good for temporal dynamics. So we then stacked and took the top 15 genes from this list. And the top 15 genes that we took, we did not just skim off the top, but we randomly selected from this green echelon because we had already looked at the data and we said, look, there's going to be correlated response. Some of these genes are just going to be doing the exact same thing. Well, we don't want to take the top 15 and find that all 15 of them wiggle the same way. we like to see some diversity in response. So we randomly indexed these, and then we just selected 15 of these from the top echelon, okay? And this is what their responses look like. So this is the actual RNA-seq data. So this is the point where, as a PI, the guess that we should try to do this $24,000 RNA-seq experiment paid off. But it's okay because we had DARPA funding, so it's just like we had to spend the money. So, but basically, like, if you look here at these error bars, these are the error bars that come from replicates, biological replicates. Okay, and so the sampling technique is very fast, right? And it requires precision. It requires attention to detail, and not all genes have a very low error bar. But what we noticed was that there was some genes that had reasonable error bars. So we were capturing dynamic temporal trends. Some of these genes had sort of oscillatory dynamics, but some of these genes were noisy, and especially noisy towards the end, which is something that we were curious about. But all of the genes, if you look here, the 0 line indicates there being no fold change response, because a 0 fold change means that the ratio of the log ratio of the malathion treated condition over the control condition is essentially 1. So then the log of that is 0. Okay, and then but basically anything that's going up has an activated effect, or is responding like an activator would, and then anything that's going down has a repression effect. So we have a mixture of responses here. The genes that we found, though, are all over the place. Okay, there wasn't one category that stood out to us. There was a collection of genes here, and it was interesting because this is a data-driven approach, and so we weren't approaching this from a biochemist perspective or even from a genetics perspective, as much as I love genetics and DNA supercoiling and all things related to genes and genome and annotations. This was very much just coming directly from the data. So you get back a collection of genes, and they tell you about the cell state, but they're to some degree all from different parts of the genome. So it raises this idea of like trying to find a summary collection of markers that you can use to determine the burden on a cell, right? To determine like if you're doing feedback control, if you're shifting the state of the cell, so it may look like you're silencing or reducing the burden in one way, but maybe you're changing the way it responds to like polyoxic shift. So these are things that could be interesting to think about. But the natural thing to do then is to take the promoter region of these genes that are identified, and the promoters aren't formally annotated. So we take a very generous region, 300 base pairs upstream of the coding sequence of the gene, and we fuse it to our fluorescent protein. And when we do that, this then gives us a reporter for that promoter. We put that onto a broad host range vector that transforms well to Pseudomonas. It uses the BBR1 origin and has a mobility element that allows it to transcribe. And this is what the responses look like of the synthetically engineered strains. So we take this plasmid now, each with a different promoter on it, and there's 15 plasmids that we built, and we transform them back into Pseudomonas fluorescens, SBW25, the wild type strain, but now with these different reporters. Then we treat them with malathion to see how they respond. And consistently we see, what I could say is that we saw a very wide range of responses, which was surprising and also wonderful because it's from a biocomputation perspective, this is fascinating. But also we saw that every sensor had its strength and its weaknesses. So some of them were better for response, slow scale response. Others were better for capturing inversion or signal inversion. Others were better at capturing short-term response. And others had both short-term and long-term characteristics. So lots that we could do here with this set. One of the things that we're currently working on, a new funded project, is trying to design combinations of these guys to make temporal encoders of chemical signals. So in neuroscience, we talk about how you can take a long temporal sequence and you can code it as a single image because you can project all of that information into one snapshot by making the data, the embedding, a high dimensional object. And what we're trying to do now is see if we can actually use the combination of these sensors to generate a high dimensional embedding that can tell you not just that a signal arrived in the environment 5 hours ago, or that it was a high dose or a low dose. That's sort of where we're at, right, with a lot of the CRISPR camera methods and toggle switches and event detectors, right, that we built. Like this is actually something that you could tell you, like, oh yeah, the chemical trace of the signal looked like this. It popped up, then it went down again, and then went down and decayed. right? It could actually give you a video recording, essentially, right? It can actually survey chemical signals. And I think that is a capability again that I think would be really cool to fill. Okay, so here's your standard transfer curve plot. You guys know transfer curves. We also tested to just make sure that if you vary the concentration of malathion, you get your standard transfer curves. No, here we're not building transcription factor sensors. All right, this is not the standard approach. This is a data-driven approach. All right, we have biomarkers. We have promoters that are responsive to the element in the Pseudomonas cell. All right, but not necessarily outside of that cell. All right, we're not trying to find a Pseudomonas transcription factor and then drop it into E. coli. We're saying, I have the non-model microbe. I want it to work in this environment. I just need to quickly find a collection of genes I can use as sensors in that microbe. And then once I found them, then I'm assuming that I'm going to stay in that context, or at least close to that Pseudomonas context. Okay. And so you can see here activators and repressors. Okay. And so the technique works certainly, and we had 15 functional sensors. The real stress test, though, was whether or not, well, so there are a couple controls that I won't show because just constraints on time. But the real test was whether or not it was specific to the chemical. Because it was clearly sensitive, but is it specific, right? Sensitivity and specificity. So we tested fructose, galactose, a bunch of sugars, then we tested permethrin, a bunch of different pesticides, and what we saw was very uncorrelated responses between all of them. That is, this collection of sensors had the capacity to differentiate all of those signals. And that became preliminary data for our next project, basically, because it was like, oh, wow, like that. The way that they wiggle is so complex that you can actually separate, looking at the temporal sequences, one sugar versus the next. All right. So these are sort of like generic cell state indicators, not quite housekeeping genes, because not many of them aren't housekeeping genes, but like they're cell state indicators that can tell you about what the cell is doing as a response to, say, the insertion of a synthetic circuit element, or as a consequence of turning on a cluster of genes that does nitrogen fixation, right? So I think there's some interesting applications here. Okay, the last experiment was really fun, guys. So I had cabbage plants that we were growing in the greenhouse, and we had a batch of 3 that were growing up perfectly. And one day I said to Akib, hey, why don't we try your sensors on real cabbage plant runoff? So we took the compound that has, or the pesticide that has malathion that's found in Home Depot. Do you guys have Home Depot here? Not really. Not really. Really? Okay. Yeah. So, but like at Home Depot, you can go and buy a bottle of Spectracide. You can't buy purified malathion. So we used analytical standards of malathion to make sure that we were really testing what we wanted to test in all of our previous experiments. But for this, we just bought dirty malathion from Home Depot. 50% of it is stated to be malathion. The other 50% is proprietary. They're organic compounds that smell terrible. Okay. So we then sprayed this chemical on all of, having established that the sensors respond to pure malathion. The question is, will it actually work in detecting the runoff of a commercial pesticide that has a malathion compound, along with all the other chemical noise that's happening? So we sprayed, 0x, 1x, 8x pesticide, and we watched basically collected all the flow through in conical tubes. And then we did size exclusion filtering to get all the dirt out and all the bark and all the stuff that comes off of agricultural waste. And then we took all of that filtrate and then we dropped it into our beakers to test for malathion response. And we found a panel of 9 biosensors, this is down selected to 9 from 15, that had some response, and 6 of them had a graded response as a function of 0x, which is just like tap water, 1x, and 8x pesticide. Okay, so this is interesting because it tells you that you can actually try to estimate like what the working concentration of spectracide is if you were to see the fluorescent signal. All right, so you could calibrate your sensors accordingly. But it also tells you that these sensors, which we found using machine learning, mathematics, control theory, actually in the end do work in a real world setting. All right. The only next level of real world is to take the sensors and then release them in the environment. But we don't do that, right? That's not in the purview of our regulatory framework. So we're not releasing microbes in the environment. We're taking microbes in the lab we're harvesting environmental samples and showing you can actually detect the compounds, okay? So all of this work is summarized in a paper that just came out last month in Nature Communications. It's called Learning Perturbation-Inducible Cell States from Observability Analysis of Transcriptome Dynamics. So, yeah, takeaways. Dynamic mode decomposition extracts critical biomarkers for biosensing, even when data resolution is sparse. That's number one. Number two, using DMD and observability maximization, we designed 9 novel performance sensors from 6,000 genes. It really is a needle in the haystack problem, okay? That can actually detect in situ runoff of a new pesticide compound that it has never seen before. All right, or maybe the organism has seen it before, but we've never tried to find the biosensors for this compound. All right. Think about what would be the other path of doing this, right? You normally have to go in the literature and you'd hope that someone has studied protein ligand binding dynamics to find a transcription factor, right? This is a different approach. It's a little bit more systems biology focused. And I like to think of the work that Tom's doing, that Guy's doing, is sort of helping to highlight, and Jose as well, that is highlighting that as we do synthetic biology, we're really running into systems biology questions over and over again, because the network that is driving the cell is really, really complex. And the touch points, all the interface points, we don't really know. We're starting to figure out what they are, but then that means there's this complex interface that you have to think about engineering. And so, you know, this is an example of where you can look at the data of a cell and you can actually identify a collection of biomarkers that are critical as a sort of interface for a perturbation. Okay, there's not much time for the second part of my talk, and I don't want to go into super detail. I'm just going to show you one slide, so we'll skip the math because that's too scary. We did the same kind of experiment here, but we did this with deep learning this time because we had more data. We were fusing fitness curves with time series RNA-seq. So we did some sensor fusion there. Sensor fusion is when you combine 2 different types of data. And I don't mean just put them in a Excel spreadsheet together. I mean, like, we actually built a model that can capture like time points from RNA-seq coming at the hour time scale versus like fitness data that's coming at the minutes time scale on your plate reader. Okay, and that's not easy to do. Then we had a neural net that was actually able to capture then this high resolution dynamics, couple it with the RNA-seq dynamics. Then we do the observable subspace decomposition, which is a thing from observability theory, and then that allows us to identify the critical genes. We then did ddCPF1 multiplex CRISPR knockdowns from the targets identified using this method, and we identified in particular the top ranked knockdowns that would give us some effect, and scored the increase in biomass or the decrease in biomass accordingly. And here's the results in summary. So you can see here the wild type, the negative control, not the wild type, which is just ddCPF1, but no guide RNA. All right, when you're not knocking down any genes whatsoever, basically this is the baseline. All right, and everything to the right on this axis right here means that in a nutrient stringent condition, a nutrient starved condition, these markers actually serve to increase the fitness of the organism. And it's combinatorial. All right. This is not normally, if you were to do this, you'd say, I need to target this gene and maybe other genes nearby in the cluster. This is like taking 3 targets that are just totally spread across the genome. It's very data driven. All right. It's a multiplex combinatorial problem. If I can pick 6 genes in the genome that I can use to alter the fitness of an organism, to increase its biomass by 2-fold, what would I pick? What 6 genes, right? It's a huge problem, right? So this algorithm is giving us a way to compute estimates or guesses that we can then test in the lab, okay? And so you can see in the nutrient standard condition, it produces very nice results. The best performing mutant is this green one right here, which combines UBID with FBP and LipA. And I would have never in a million years thought to combine those 3 genes, all right, in guide RNA form. But that's certainly what this algorithm returns. One that's dear to my heart. So the mutant that consistently showed that you could actually knock down fitness if you want to like control an organism and make sure it didn't grow too fast is TOPA. TOPA is an enzyme that, so you can see here, it's reduced fitness compared to the wild type. So when you knock down TOPA, the cells stop growing as fast. And that's actually consistent with our understanding of supercoiling theory, right? Topoisomerase A is the enzyme that is used to maintain negative supercoiling in DNA. So it relaxes the DNA and it enables transcription across the genome. So if you knock out that enzyme, you're basically going to shut down transcription across the genome consistently, right? And remember, we're not knocking out, we're just doing knockdowns. And so that's enough to then create this fitness defect, which is a nice sanity check. Okay, so these are the 2 papers. This one's on Nature Comms, this one's on Archive. It's currently under review at Automatica. We have another paper that's in the work that describes the experimental work, but that's coming out a little bit later. I just want to shout out a quick plug to some of the new projects that are happening in our group, and just and shamelessly plug for if you guys are interested in opportunities. So Tom probably knows my work from my supercalling days, and I built different versions of toggle switches and different versions of CFP and RFP that were in different orientations, and discovered alongside Jim Collins and Richard Murray and Domitilla that basically how we were laying out genes was creating massive interference patterns in how our circuit parts are responding. So when I started my PhD, I was like, I want to quantitatively model every single part. And then what I discovered at the end of my PhD was this variable that was affecting every single part that we were trying to quantitatively model. All right. And so now I'm still trying to understand this phenomenon because it's a really beautiful, complex phenomenon, right? If you think about DNA, it's a double-stranded molecule. And when you transcribe DNA, basically it releases positive supercoiling, or sorry, propagates positive supercoiling downstream of the transcription bubble and negative supercoiling upstream of the transcription bubble. Now, classical models say the transcription bubbles just move along the DNA, and they're 17 base pairs in length, and then as they move, basically the DNA kind of relaxes and then rewinds. All right. The problem is that people have done a lot of studies showing that multiple RNA polymerases actually bind to a single coding sequence, and so there isn't really an opportunity for a DNA strand to fully relax. Fast forward to operons. When you look at operons, we look at the transcription profiles from pile diagrams at a snapshot time point from RNA-seq data, and you see thousands of base pairs right through the terminators, right through the promoters, all just being transcribed. Okay, which suggests again this idea that DNA oftentimes will assume a very relaxed state in some regions for thousands of base pairs, but then those twists in the coil will go elsewhere. All right, so we have methods in the literature since the 50s and 60s using quinolone gels to measure the density of twist in DNA at a global scale. If I take a plasmid, I can say, what's the overall twist in the plasmid as a function of how fast it runs through a certain kind of chemical gel. But we don't have any way to model or to actually measure localized twisting and writhing. Localized twisting and writhing in the sense that downstream of this gene, not as a property of the whole plasmid, is there a buckling event? Is there some or not that's forming? All right. And so I recently have an NSF Career Award that's just started that is on this question. Sorry for the typo. So it's a novel framework for measuring and engineering twisting and writhing in DNA. And then secondly, we have an Army Research Office grant that's starting that is basically looking at how do we use the insight from this project and the principles that we learned to design plectineme or not insulated genomic integration sites. And this project was very much inspired by our work with Chris Voigt, where we were looking at integrating fragments of DNA into the genome of many bacteria and discovering that we were seeing a regularity in how bacteria responded, just as a same sequence of DNA was integrated over and over again. So the rig that we're building right now, and that we've had some moderate success with early on in our lab, is one that will combine the technology of cell-free extract. Being a former student of Richard's, of course, I'm very interested in cell-free extract as well, and magnetic tweezer technology to be able to take a strand of DNA, functionalize one end of it with a magnetic bead. The other end will have a cognate antibody that will then bind to a capillary tube or to bind to the glass surface of some surface, and then will flow cell-free extract through. Now, as we flow the cell-free extract through, what happens is that this DNA, if it has a promoter, will actually become transcriptionally active. Now, previously, the Decker lab in the Netherlands showed that if you take DNA that was not transcriptionally active, and you just manipulate it with magnetic tweezers, you get the DNA to contract and shrink and buckle, and you could visualize it. So we want to do this while simultaneously doing transcription. And so here's our first experiment, a proof of principle experiment on this project, where we took magnetic tweezers. We rotated the magnetic tweezers after we saw an initial burst of M. broccoli fluorescence, so an RNA aptamer. And then after we induced the magnetic tweezers, we actually entered and created a second phase of expression in which the RNA sort of took off. And so, so this is a project that I'm interested in. We're looking for a PhD student and 2 postdoctoral scholars. So if you know someone that's interested, or if you're interested, feel free to reach out. So again, big thanks to Akib and Shara, who were the 2 drivers of the work that you saw today. Also, all of the wonderful students in our group and the lab alumni that have helped throughout the time, through the time that they're here. and then of course our funding sources. Thank you so much, Guy, for the opportunity to speak.